results. Now we shall proceed with the next event on our stage, and we have two participants at the discussion which will participate virtually. Dr. Björn Heindryckx, you know him already. Dr. Aivita Putnina, she is the head of Department of Anthropology at the Faculty of Humanities, University of Latvia. We should see her any moment. Also, in person, Andris Grunskis. Andris is the head of embryology laboratory at the company IVF Riga here in Latvia. And the discussion will be led by Dr. Inese Čakstiņa. Inese, welcome. So nice to see you here.
Thank you.
She is leading researcher at Riga Stradiņš University Institute of Oncology, molecular biologist by education and the mindset. So, ladies and gentlemen, here we go. Inese,
Thank
the
you,
floor is yours.
very, very much for the introduction. And I think Professor Björn Heindryckx already gave a really beautiful introduction into this topic. A decade ago, or even less than 5 years ago, when I was giving the lectures about the gene therapies, I was talking about the two main types: the gene therapy or gene engineering in somatic cells, and gene engineering in embryonic cells or germ cells. And back in that time, I said, like, okay, this gene therapy possibilities in germ cells is just only theoretical because we do not have the technical possibilities for that. However, now we already can see that the technical possibilities are arising, especially because of this CRISPR-Cas system. And I wanted to make this discussion in a way that we start to talk with the more technical part, and then we're going to move to these ethical parts. And the first question would go to embryologist Andris Grunskis. Andri, maybe you can explain how easy is it to manipulate a very early embryo, and what should people take into the account, like how early you can actually touch this embryo for the manipulations?
Hello. Actually, you can start as early as from the first day, as Björn showed in his presentation. As soon as you get oocyte, you can manipulate oocyte, or I think the best stage to do it would be the zygote stage, because then you have both genetic materials from both parents, and then it's like I know it's like a driving car. When you first do it, you think it's impossible to do it. You don't know where all the clutches are and the buttons to do and what to do on the road. But if you have experience with the daily ICSI and trophectoderm biopsies, then it's easy. You just need materials, and if geneticists or the persons who are responsible for preparing that, they give you the tools, then you can just insert them where they're needed. So it's quite easy, I think, from the practical part.
Mm-hmm. Why not use the germ cells themselves? Why not do the gene editing on oocyte and in sperm cells separately?
Well, you could try to do that on oocyte, but I think it's quite impossible to do it on spermatozoa, because you're gonna insert the tools, the CRISPR-Cas system. There's no cytoplasm to put that.
Mm-hmm.
So I think it's the best, as I said, and then as it was shown in presentation, that the best stage to do it would be zygote stage.
Mm-hmm. Yeah. Björn, I had a question for you. You already mentioned this problem of the mosaicism, and also you showed one possible solution for that is injecting as early as possible after the fertilization. Why don't people do that now more and more if they know about this problem, or is this the way where this editing goes into?
Yes, that's correct what you say. People have seen that the earlier you do it, the better, because the CRISPR-Cas tool has to do its job, and it has to do its job before the embryo starts to cleave. Otherwise, you will have cells that have not been edited and cells that have been edited. The problem is, you have to do it. We are now sure in human, you have to do it at the stage of fertilization. So when you bring together the sperm with the oocytes. So when you inject the sperm into the oocyte, you simultaneously have to inject this fluid, which contains the CRISPR-Cas tool together with sperm into the oocyte. So why people are not sometimes doing it, it's because they are not allowed to make embryos for research purposes. So it's only in a few countries, like in the US, it's not allowed to make, in some parts it's not allowed to make embryos. Also in Europe, there are only, I think, 3 countries, if I'm correct, that can make embryos only for research purposes. So it's Sweden, UK, Spain, and Belgium, so 4 countries. So yeah, there is also a lot of ethical and legal constraints that people have to try to target. A lot of countries do allow to do research on available embryos. So we have embryos available for doing research, which are not used anymore for the patients. But it's very difficult to get this CRISPR-Cas tool in all the cells of the embryo. You will end up with much more mosaicism. So it's a little bit practical, legal and ethical constraints that people are not doing all on the fertilization stage at this moment.
In your presentation, you mentioned also another one, I would say, quite large risk using the CRISPR-Cas technologies, because I also remember from 2017, there were already appearing the articles showing that as much as we wanted this to be very precise molecular scissors or editing tool, there are still some problems, and sometimes people can observe some site-unspecific editing. Especially, it's really concernable, and people are concerned about waking up some proto-oncogenes, thus raising the oncological events into the cells. Do you have, this would be a question for the Björn and also for Andris, like how to deal with this part, and for Andris it would be like how easy or how much more expensive it would be to test those embryos after that. I think we can start with Björn.
Yes, about off-target issues and on-target, if it really works what you want to modify. Well, the bioinformatics also are developing it, so this is also helping us to increase the efficiency. So actually for the embryo gene editing, we don't see so much unintended off-target things that happen. And also, people do a lot more research on somatic cells and stem cells to do this gene editing. But the mechanisms of repair in stem cells or somatic cells versus embryos, it's different. We cannot compare it. So we still need to get more knowledge in this. But of course, also what is important are techniques for detecting off targets, or the efficiency in on target have developed also tremendously. So now it's all sequencing, and we can do a lot of detailed check if it's all working correct or not. But my honest opinion, the CRISPR-Cas tool, the classical CRISPR-Cas technique, it will not be very efficient in human embryos. There are more and more papers coming up that we have to shift towards more modified version of the CRISPR-Cas gene editing tools, especially in the embryos. For somatic cells and stem cells, the classical one is working quite efficiently, but for embryo gene editing, we will have to develop newer techniques of CRISPR-Cas.
Andri, you are working as actual embryologist, and you know all these steps that the embryologist has to do from the moment of the fertilization until the moment of the implantation in the woman's womb. If there will be such a technique to modify the embryos, you would probably expect a lot of validation methods. How much burden it would be for example, IVF clinics and embryologists?
Well, I don't think that there will be much of a changes, except the one that we have to inject the CRISPR-Cas system at the time of fertilization, because now already today we're doing a lot of embryo biopsies, and that's it actually, because you do the injection of the modifying tool, and then you take the biopsy to check whether it had worked. So it's because nowadays we do a lot of PGTA or PGS, and then you use the same material.
So when you use the biopsies, you use just only few cells. How can you be sure that there will be no mosaicism?
Well, you can never be sure. Well, that's a simple answer.
So this is one of
Yes.
yeah.
Yes, but people are working, and also in our lab we are working on this to check in a research context. Yeah, we are all doing it in a research context. So we compare our trophectoderm biopsy with the rest of the blastocysts, all cells, and we check now if it's really representative for the whole of the embryo of the blastocyst. So these are things that really need to be sorted out before we ever think of any clinical application. That trophectoderm biopsy is indeed representative for the whole embryo.
Thank you. I wanted to introduce in our discussion Professor Aivita Putnina. So, probable society with less genetic defects. What could we say about this possibility from the viewpoint of the anthropology and the humanitarian sciences?
Well, as it has been discussed, there are probably more implications to that what already had been mentioned, because we can really read and check gene sequence and we can edit it, but the trouble is that sort of meaning to these letters is culturally attached and culturally created, and there is really sort of slim line sort of not to get into eugenics, which was also one of sort of dreams of early 20th century. And we see that this attitude and regulation already of the gene editing technologies is quite controversial, and states, national states have really different solutions in allowing to use this technology. And yeah.
At the end of Björn's presentation, he showed that there are some laws in Belgium that allow to do some research, but they are avoiding the research that would be driven towards the eugenics, like to change the eye color or whatever for the baby, how it looks. So can we talk already in those two categories that society would be more acceptable for the gene editing with the purpose of the medical implications, yes, to cure or to avoid some genetic defects, and a little bit less acceptance for the designer babies? What is the society?
Here, yes, the trouble is, so what is sort of the definition of effect is essentially social. And the context probably most controversial is this germline editing. And this editing probably is already, sort of these ethical dilemmas, they already have arisen with the sister practical technologies in general. And this is a context. So it's the same technology, but it exists in these two social contexts. One is assist reproduction technologies, another is this genetic editing of somatic cells, which is more acceptable because this is interpreted in line of different already available therapies. So this germline editing, it's more problematic, and we know that even, for example, pre-implantation diagnosis has been also quite controversial. Just to mention, for example, Duchesneau and McCulloch case. This was a couple who wanted their future baby to be deaf. And in this pre-implantation diagnosis, they wanted that this child with a faulty gene, which would cause deafness, would be implanted in later born because they were both deaf as parents. And this also created controversy whether we can decide for this and can we edit for certain medical conditions which still produce livable life. And where is this sort of burden of illness? So we have genetic screening against Down syndrome, for example, and we can make medical abortion and select these conditions. But this again is a really ethically dubious thing, and where it draws a line, what would be that condition? And what makes it even more problematic, because this sort of standard for what we aim at is, again, culturally decide autonomous, self-sufficient individual.
What do you think, like, who should decide the line which should not be overstepped in the research and probably later on implementing into the life? Because as we can see, back in the 80s, there was quite a lot of already concerns about the IVFs, those reproductive medicine parts, but nowadays the IVF clinics are almost everywhere, and it's the common practice. Also with the embryonic stem cells, when they first appeared, there were lots of concerns. Nowadays, still the concerns, but the embryonic stem cells can be obtained and at least for the research can be used. As we can see that somehow the society slowly but kind of moves forward. So in the imaginary scenario, whenever, probably, if we were going to get to the point where these genetic engineering, these genetic modifications will be allowed on some certain level, what do you think, who should be the one who decides? Either it's parents or governments, or what should decide if it is allowed, if it should be allowed or not?
Yeah, I'm afraid that there is no answer, and because it's really a question of responsibility and whose responsibility. We in Western societies we favor individual uniqueness and autonomy. So if this uniqueness is being interfered with, so who decides for this? Is it parents? So in a way, parents decide not about their own body and treatments. They also decide upon genetic makeup of their child. So in a way, this person born had never given any informed consent. And this is also troubling with our cultural concepts. Also, it creates threats for responsibility. So we also made research, and a lot of parents in Latvia, they feel responsibility for banking stem cells. So just in case their child would need some kind of a stem cell treatment in future. So in a way, it also expands our responsibility. And this is also not really neutral, because it's, and this is really present in evaluating also CRISPR technologies, is it would probably increase also social inequalities. And this is also a question of equity and justice and accessibility of technologies to all. So this makes this answer really difficult, and we might use citizen panels to decide on this, but until these questions of equity are not answered, it also will be a really difficult problem to decide, I think.
Thank you. Björn, maybe you can comment a little bit on this as well, how it is in Belgium, because you already have some laws about it.
Yes. Well, I don't think we should let it depend only on the patients. Yeah, some patients really have their own very high wish to have their own genetic child, and they would take a lot of risk, even if the technique is not 100% efficient or safe. I know a lot of couples would already say, oh, try it. It's an opportunity to get a healthy child, try it. So if we only let it depend on the patients, that would not be a good idea, of course. So it should be an interplay between clinicians, of course, play an important role, but also the government. There should be new laws implemented European-wide, for example, that regulate this, especially this gene editing in the germline. And there are also some societies like this Eschery Society who can help in this making up these regulations or recommendations. But also, the government should play a role in this and implement new laws, because the laws are already dating, like this Oviedo Convention, and in a lot of laws in countries, there is nothing mentioned about gene editing in germline, because it was, it's still a new thing. And also the public has an important role. So as Aivita was saying about this, we should also do surveys in the public to see how far we can go, because as scientists we can go far, but is the society also ready for this? So in Belgium we are very actively waking up the public and trying to do surveys and open discussion on this topic, because it will come one day. We will do it one day. Not we or other people will do it one day, like this Chinese guy has done it, unfortunately. But we have to open the debate, and there are a lot of good organizations and committees who can really make up very strict regulations and guidelines what to do in the future. So I would encourage that this still is happening a lot, especially about this gene editing in the human germline.
So we can say that for the scientists, this gene editing, this CRISPR-Cas is almost like not anything new, because it's already about 5 years old. And we know that the public and the society is always very cautious about the new things and very kind of drawn against the new things. You mentioned that you are doing the public surveys. Do you have at least some results? What's the public's attitude towards these news?
No, we don't have, we're just starting this, but I know, for example, there was a study recently published in Japan from a Japanese survey, and a lot of people were in favor of doing it, especially in a preclinical context at this stage. But so it was mentioned, and I have to be honest about this, a lot of public also acknowledged that they didn't understand fully the technique and the possibilities. So we have to work on making the public aware of what we are doing, what are the possibilities, and so on. So it's still all very in its infancy, and I hope people invest time in also getting this public opinion of what is possible or not.
Björn, I have another one question for you. I know that when I was working with the embryonic stem cells, there was this law or the rule that you can go on only until the day 14 with those cells. How is it with the CRISPR-Cas modifications when you modify those embryos? How long can you grow them? Probably not until the birth, but how long it's allowed to kind of cultivate them?
Yes, we still have a worldwide ban on extending embryo culture until day 14. So we can do some manipulations in embryos, but we still have to stop growth of the embryos at day 14. That's still a rule. But now, people are also making these artificial embryos, so not made by a fusion of a sperm with an oocyte. We can also now make embryos just coming from stem cells. And people are now thinking or trying to shift this deadline 14 days, because it's an embryo not coming from a sperm or an oocyte, it's only coming from stem cells. So now there is a discussion going on of these artificial embryos. Should we still consider them as normal embryos? And if they are not considered as normal embryos because they cannot grow to a new individual at this moment, can we bypass this stage of day 14 to get more information about gastrulation and so on. So it's also a very evolving field that I'm curious to see what will happen there.
Can we use the animals? That would be probably a little bit less controversial.
Yes. Yeah, that's a good remark. We still need to invest also in mouse models. Yeah, people are now shifting. Yeah, it's a competitive field. They always, if you do research in anything, people try to shift more rapidly to the human because, you score higher. You get more articles in Nature and Science in the big journals if you have results in human embryos, of course. But yeah, people are a little bit neglecting the animal work, and that's indeed a good remark. We are still also investing a lot in mouse models because, in mouse models, we can transfer back the embryos, we can see what's happening in the offspring. But people are tending, to shift too fast to human, and we have other animal models which are more relevant for human, of course, like the monkeys. So I would also encourage that we still invest also in animal research before think of clinical application, because in animal models you can transfer back the embryos and see if something if it's all safe and efficient. So people are a little bit they want to be the first in everything in human because we should not indeed forget animal experiments for this research.
Yeah, thank you. And in the last 8 minutes, I wanted to ask each of the people here in this discussion, and to summarize, like, what would be, from your point of view, what would be the pros and cons of the genomic engineering of the embryos? Maybe we can start with Andris, like some summary.
Well, the pros definitely would be if we could eliminate a lot of genetic diseases or that otherwise would be like incurable, and then if we could allow a lot of patients to have their own biological children, and then that is a big issue for people trying to conceive, because a lot of patients, for example, they don't have they have a really difficult time to switching to donor gametes if they don't have their own or they are defective. So I think the most beneficial would be to cure diseases. Yeah.
Do you see the future of the CRISPR-Cas editing or genomic editing in embryos?
Well, sure. If anyone has seen the movie called Gattaca, then I think we're in the beginning of that, because the movie is about the family that has one child born naturally, and he has defects, and there's this science space program where only the perfect people can apply. So he's falsifying his identity to be this perfect human. So it's I think there's the dilemma of ethics and that's the thing. So.
Thank you, Björn.
Yes, I agree with Andris about this. So it should be in the first phase only used to correct medical conditions that we cannot solve with PGD at the moment. Of course, if it's all working efficient and safe, we can increase the number of embryos available for these patients, because if we now do PGD, only 25% of the embryos is not bearing the mutation, or only 50%. So if it's all working 100% efficient and safe, all the embryos will be available for transfer, and these patients only will have to do one cycle to have a healthy child, for example. Especially for medical reasons at the first point. But especially, it's also very handy too to do a gene knockout. So we still don't understand a lot of human embryo development, which genes are responsible. So for example, for embryo development, we have some patient couples where the embryos arrest at a certain stage. So to unravel all this molecular network, it's a very nice tool to have in our lab to knock out the gene and to see what's happening with the embryo development. So for research purposes, it's a very nice tool. I don't think we should go in genetics, and that's of course a main problem of this Chinese doctor, that he did it actually for eugenetics. So he wanted to create children which are immune for HIV infection. Only the fathers were affected by HIV, but we have techniques in our IVF lab to wash away the high HIV virus and to make babies which don't have HIV. So he was doing it already for eugenetics, and that was, of course, not a good choice of him to do this for these couples. If he would have done it for an actual medical reason, it would not have been so controversial.
Aivita? The future of the CRISPR-Cas and the problems, probably.
Yeah, I think that genetic engineering in stem cells probably will proceed without much problems, but genetic engineering in germlines, this will be problematic, and firstly because of cultural reasons, and one of them is certain medicalization and genetization of life, which even it's kind of seems really scientifically neutral. You can imagine it as scissors or surgery or syringe injecting sort of things, and it kind of gives this light impression, but it also modifies our understanding of life and nature, and it also interferes with our understanding of and use of kinship or parental autonomy and many other things which currently create basis of our life. So even similarly as was cloning, which is technically possible, I think that these technologies will remain controversial due to these different implications. And if we say that it's for medical reasons, these medical reasons are also socially, culturally defined. And we know that in human evolution, our caring for each other and sociality had played a crucial role. So in a way, we want to evolve as more autonomous, independent beings, which is interesting extension of our Enlightenment project.
Well, thank you very much. I think it was really interesting discussion. What I wanted to remind to everyone who listens that very often we think that something is caused by only one thing went wrong. However, very often in our lives, it's not only one gene that went off or works in or is defected. So right now, the scientists and research is driven towards using the CRISPR-Cas system and using these genetic modifications towards the repairing this one gene that could cause this specific disease. However, in the future, especially if we are talking about the designer babies, already as in Björn's presentation, we could see our eye color, our skin color, and lots of other things like our intelligence is not defined by only one gene, and it will going to be the clusters of the genes. Also, I think there will be, we have to look forward how it will going to go in this area, whether it will be allowed to grow the embryos longer than 14 days, because as we know, when the organism is developing, there are lots of interplays between lots of genes. And we do not know, I think, at this point yet enough, like this 100% what each gene is doing. So I think in the future we have a lot of fundamental and, basically the fundamental work in researching the genes. So maybe any final remarks from our debaters? Andris?
Well, I think we briefly covered like the whole thing. Well,
I tried.
Yeah.
Björn?
Yeah, I think your last remark was very valuable, and it's especially for the public to understand that, it's not so easy. Yeah, sometimes people say you can do it in your garage. We are already doing it for 2 years, and now we just come up with the first results of correcting a very easy mutation. So some mutations are more difficult to repair than other mutations. So it's not like that. You need to have embryology skills, but you also need very much genetic skills to check out everything. And indeed, all these morphological traits, that will be very, very far future. We still don't understand all the genes that are responsible for this, so we should not be afraid at this stage for this.
Aivita, final remarks?
I think that public generally is not skeptical to these technologies, and I think this gene editing is starting with really kind of good and considerate PR compared, for example, to genetically modified plants, which started really with a wrong foot. So I really hope that on the one hand, this is also exciting way of, at least for anthropologists, to examine culture and also to cross-cut such cultural notions, which we have considered really sort of taken for granted. And for that matter, for example, is disease.
Well, I think there are so
is-
many, I think there are so many still open questions and we can debate and debate. Unfortunately, our time is up, and I thank you very much, everyone who participated in this debate. Andris Grunskis, embryologist, Björn Heindryckx, professor of regenerative medicine, and Aivita Putnina, our humanities representative. Thank you very much.
Thank you.
Yuri,
Thank
it's
you.
all yours.
Thank you, dear colleagues, and Inese, you in particular, for jumping in as a moderator today.
Thank you.
And to close it up, this discussion, we shall return to our survey, ladies and gentlemen. We asked you to pick the word that describes your feelings the most when you see the combination of words genetic engineering. And the results are the following. Possibilities gets 40% of the answers, responsibility even more, 50%, scaring takes 10%, and the answer the inhuman has got no answers at all. It doesn't surprise me very much, since the audience is more or less people interested in life sciences. But to round it up, ladies and gentlemen, believe me, in a couple of hundred years, we shall be divided on this planet into two big parts, those who are genetically modified and those who are not. I think so. Anyway, thank you very much, dear colleagues. We make a break now.